There is a woman who is the ancestor of every human being alive on Earth today. Not in the sense of mythology, not in the sense of religious tradition, though the name scientists gave her inevitably evokes both. In the strict, measurable biological sense of population genetics, every person alive, every man, every woman, every child on every
My
continent
children, come here.
traces their maternal line back to a
I
single
missed you
woman who
too.
lived in
Come
Africa
sit.
somewhere between 150,000 and 200,000 years ago. Scientists call her Mitochondrial Eve. And the story of how she was found, what she actually was, and what the gap between her and Y-chromosomal Adam reveals about the deep history of our species is one of the most profound and most misunderstood findings in the history of science.
I've walked through many storms.
The first thing to understand about Mitochondrial Eve is what she was not. She was not the first woman. She was not the only woman alive in her time. She lived in a world that already contained thousands of other women, each of them fully human, each of them our ancestors in many other respects. She is not the ancestor of everyone in every biological sense. She is the most recent common ancestor through one specific line only, the unbroken chain of mother to daughter
We
to
walk
mother
the ancient
to
path,
daughter that
strong
connects
and
every
united.
living person
Through
to
the
a single
golden
point
light,
in the
our
past through their
spirits rise.
mitochondrial DNA.
Forward we go with every
The
step.
mitochondria are tiny
My
structures
people,
inside
listen.
every human cell
A
that
great
generate the
danger
energy
is
the cell
coming.
needs to function.
We must stand
They have
together.
their own DNA,
Our
separate
ancestors
from
will
the
guide
DNA in
us.
the cell nucleus. And that
See this plant?
mitochondrial
It has strong
DNA is
medicine.
passed from mother
do you use
to child
it?
essentially
will
unchanged
show you.
across
We
generations,
take a little piece.
accumulating small
It's
random
beautiful.
mutations at a measurable rate. Because it passes only through the maternal line, it carries a pure record of maternal ancestry that is not mixed or diluted by paternal contributions. When scientists compare mitochondrial DNA from people around the world and trace the mutations back to their source, following the branches of the maternal family
This
tree
stone
backward
is from
through
our
time,
land.
This
all
is
of
your
those
graduation
branches eventually
certificate.
Thank you,
converge on a
grandmother.
single point.
I will make you proud.
Remember
That point
our
is
stories.
Mitochondrial Eve. She is the woman at the root of the tree. The discovery itself is one of the most intellectually dramatic moments in the history of modern genetics. In 1987, a team of researchers led by Alan Wilson at the University of California, Berkeley, published a paper in the journal Nature that compared mitochondrial DNA from 147 people representing populations from around the world. The analysis found that all of the mitochondrial DNA in their sample could be traced to a common ancestral sequence, and that this ancestral sequence was most likely African in origin. The tree of human mitochondrial diversity, the paper argued, was rooted in Africa, and the woman at that root had lived approximately 200,000 years ago. The paper caused an immediate sensation, not entirely for the right reasons. The name Mitochondrial Eve, which the researchers did not themselves use, but which appeared almost immediately in popular accounts of the finding, suggested to many readers that science had confirmed the biblical creation story. That there had been a single original woman from whom all humanity descended in the theological sense. This was a misreading, but it was an understandable one, and correcting it turned out to be harder than the researchers had anticipated. The original paper had made its argument with the careful qualifications that scientific papers require. The popular coverage stripped those qualifications away and replaced them with a narrative that was simpler, more dramatic, and more theologically resonant than anything the data actually supported. The resulting confusion was substantial enough that several of the original researchers spent significant portions of the following decade writing follow-up papers and popular articles specifically to address the misinterpretation. The correction was important not because the theological reading was offensive to the researchers, but because it obscured the actual finding, which was considerably more interesting than the theological interpretation it replaced. The misreading persists in popular understanding today, and it obscures something that is, properly understood, far more profound than any simple origin story could be. To understand what Mitochondrial Eve actually was, you have to understand what it means for a lineage to go extinct without the individual going extinct. Every woman alive in Mitochondrial Eve's time had mitochondrial DNA of her own. Some of those women had daughters. Some of those daughters had daughters of their own, and the mitochondrial lineage continued. But some of those women had only sons, and their mitochondrial DNA was not passed forward, because sons do not pass mitochondrial DNA to their children. And some of those women had daughters who themselves had only sons, and their lineages also ended. And some had daughters who died young, or who had no children at all. With each generation, some mitochondrial lineages were extinguished, not because the women who carried them were in any way inferior or unlucky in a broad sense, but simply because the specific statistical lottery of reproduction, of who had daughters and how many, gradually eliminated all lineages except one. The one that survived was the lineage of Mitochondrial Eve. It was not the oldest lineage. It was not the most diverse lineage. It was simply the lineage that happened, across 150,000 years of random demographic processes, to never fail to produce a daughter at every generation. This is the concept that population geneticists call lineage sorting, and it is the mechanism that produced Mitochondrial Eve in a world that contained thousands of other women. Given enough time, any population will sort its mitochondrial diversity down to a single ancestral lineage, regardless of how diverse that population was at any earlier point. The woman at the root of the surviving lineage becomes, by definition, the common ancestor of everyone descended from that population. She did not earn this distinction. She did not survive when others did not. She simply had a daughter, who had a daughter, who had a daughter, in an unbroken chain that continued while all the parallel chains gradually died out around it. What the 2013 discovery of Y-chromosomal Adam's true antiquity revealed, when placed alongside the estimated age of Mitochondrial Eve, is a temporal gap between our common maternal and paternal ancestors that forces a fundamental reconsideration of what early human populations looked like and how they were structured. Mitochondrial Eve lived approximately 150,000 to 200,000 years ago. Y-chromosomal Adam lived approximately 200,000 to 340,000 years ago, and the 2013 lineage pushed that estimate even further back. The gap between them is real, and it is large, and it is not an error in the analysis. It is telling us something specific about the social structure of early human populations. The leading explanation among population geneticists is that early human societies were structured in ways that created different demographic pressures on the male and female lineages. In populations where women tended to remain in their birth communities, while men moved between groups, perhaps through patterns of male exogamy or through the specific dynamics of male-male competition for reproductive access. The Y chromosome would have experienced more severe bottlenecks than mitochondrial DNA. Successful males might father large numbers of children, rapidly concentrating Y chromosome diversity in a few lineages, while female lineages remained more diverse, because women's reproductive contributions were less concentrated in a small number of individuals. Over hundreds of thousands of years, these different demographic pressures on the two lineages would produce exactly the pattern we see in the data: an older common paternal ancestor and a more recent common maternal ancestor. Because the paternal lineage was subject to more frequent and more severe bottlenecks that reduced and re-reduced its diversity, while the maternal lineage maintained greater continuity. This is the picture that emerges from placing Mitochondrial Eve and Y-chromosomal Adam side by side and asking what their temporal separation means. It is not a picture of two ancestors who missed each other across time. It is a picture of a social world in which the biological experiences of men and women were so different, in terms of who reproduced, and how many offspring they produced, and which communities they belonged to, that the genetic records they left behind look like they come from different histories, because, in some meaningful sense, they do. There is a moment in the 1987 paper's reception that captures something important about what this science asks of us. When the initial news coverage described Mitochondrial Eve as the mother of all humanity, the researchers spent years trying to correct the impression that they had found the only woman alive at the time. She was not the only woman. She was simply the woman whose maternal line happened to survive. All the other women who lived alongside her were also our ancestors in many other senses. Their nuclear DNA is in our genomes. Their cultures shaped the world that Mitochondrial Eve's descendants inherited. Their children and grandchildren were the people who kept the human population going through the demographic challenges that the period presented. They are not erased from our ancestry. They are simply not visible through the specific window of mitochondrial inheritance. Mitochondrial Eve is the answer to one question only. Who is the most recent woman from whom all living people descend through an unbroken line of mothers? That is a real question with a real answer. But it is a smaller question than the one most people think they are asking when they invoke her name. The woman herself would not have known she was special. She lived her life in a community of people who were, in all the ways that mattered to daily existence, her equals. She gathered food or prepared it. She had relationships and conflicts and moments of joy and grief that left no trace in any record, except the mitochondrial DNA she passed to her daughters. She watched her children grow and her parents age. She had no awareness that the particular chain of daughters that happened to descend from her would persist through ice ages and migrations and civilizations rising and falling and catastrophes of every kind. For 150,000 years and counting, until a team of scientists in a laboratory in California would one day look at genetic data from 147 people around the world and find her at the root of the human family tree. The one woman whose maternal line had never once failed to produce a daughter across a span of time so large it resists ordinary comprehension. She was not the mother of humanity in any complete sense. She was a woman in Africa, living her life, doing what humans have always done. The fact that her mitochondrial line is the one that survived tells us something about the randomness of deep time. About the way biological history is written not by the most significant individuals, but by the luckiest chains of daughters in a world where most chains eventually end. And the fact that we can find her at all, that the genetics can reach back 150,000 years and place a finger on the root of the maternal tree, tells us something about how much of the past is still present in every one of us. In the mitochondria of every cell in every human body alive today, carrying a record of maternal descent that no historical catastrophe has ever been able to erase. The science does not make her divine. It makes her something more interesting than divine. It makes her real, a real woman, in a real place, at a real moment in history that we can now approximately locate, who had no idea that her daughters' daughters would one day give rise to every human being alive. That is not a religious story. It is something better. It is what actually happened.